Hi all, welcome to SL Biologica, a foundation to license, which satisfies all your license cravings. And I'm here to discuss with you some of the important aspects of immunology in the form of MCQs and its associated discussions. And this would be really helpful for your PhD level competitive exams preparations like CSIR, ICMR, ICAR, DBT, and GATE, as well as for your PhD exams. So let's move into the questions. The first question is actually it's related to the phagocytosis. We will move to the question. Which specific sequence correctly describes the key steps of a phagocytic cell eliminating a pathogen starting from ingestion? So they are asking about the correct sequence of steps that is actually happening with the phagocytosis. So let's see what is the phagocytosis process or how the process is happening. So it involves forming a membrane vesicle that is known as the phagosome around the ingested pathogen. Okay, and this phagosome then fuses with the lysosome of the cell, which is having various lytic enzymes like the hydrolytic enzymes to form the phagolysosome, where the digestion happens, and these digested products are then released to elicit the immunological response. So here we have the sequence of events happening starting from phagosome development, then it fuses with the lysosome to form the phagolysosome, then digestion happening, and finally release of the breakdown products. So now let's see the options available here. The first option is pseudopodia attachment, phagosome, lysosome, and then digestion. So we know that this is not the sequence happening. Okay, and second option, it starts from phagosome. Yes, phagosome, it starts from the phagosome, then comes to the lysosome fusion to form the phagolysosome, and where the digestion happens with the hydrolytic enzymes of the lysosome, and these digested products are exocytosed to elicit the immunological response. So this represents the correct sequence of events that is associated with phagocytosis, right? So the correct option is actually B. However, let's also discuss the other two options. C, it starts from lysosome, then phagosome, digestion, then release of the lytic enzymes. So this is not the way how the phagocytosis is happening. It never starts from the lysosome. We know that now. And the last option is starting from endocytosis, then phagosome, and then TCR activation. So this is also not the correct sequence. It eliminates the involvement of lysosome, right? So the correct sequence of events is represented by option B, starting from phagosome, lysosome fusion, then digestion happening within the phagolysosome to digest the particles, and then finally exocytosis to release the products. So we have the correct choice is B. So here is a pictorial representation of the phagocytic events happening. Here we have the cell, the bacteria, which is actually interacting with the surface receptors of the antigen presenting cell, I mean the phagocytic cell. So here you can see the receptors. Once it interact with the bacterial surface receptors through endocytosis, this bacteria is getting engulfed, okay, to form the phagosome, and this phagosome will then fuse with the lysosome to give the phagolysosome. So the phagolysosome is having the lytic enzymes of the lysosome as well as the bacteria entrapped within that, okay. And within the phagolysosome, this lytic enzyme acts on the bacteria to digest the products, and once it is complete, once the digestion is complete, this then get released to the external compartment through exocytosis, and this can then further elicit the immunological response. So this is how the phagocytosis process is happening. Okay, so now it's clear, right? So let's move to the next question. We have the second question. Which among the following best describes the outcome of a clonal selection? So they are asking about the clonal selection process of lymphocytes. Okay, the choices are: first one, elimination of all lymphocytes reacting to any antigen. Actually, from the term itself, it is saying about clonal selection. So there is an involvement of a choice. We are selecting something. Okay, so keep in mind that thing while we examine the options. So the first option says about eliminating all the lymphocytes reactive to any antigen. So that doesn't represent any choice, any type of selection in that. It is eliminating all the type of lymphocytes which is reactive to any antigen. Okay, so that shouldn't be the answer. Okay, so the second choice is, the second option is activation of all the lymphocytes simultaneously. Here also we can't see any kind of choice or selection happening. So the third choice is expansion of specific lymphocytes recognizing the antigen. So some sort of choice or selection is happening here. It is represented by the third option. Okay, let it be the fourth choice is induction of non-specific inflammation. Okay, so we have in our mind that there is a chance for this option C to get correct because it is involving some kind of choice or specificity or selection process that is happening. So before going to the correct answer, let's see what is actually clonal selection. So we know that each of the lymphocyte is having a unique receptor. Okay, so once the antigen is there, this then bind with the specific receptor. So this antigen selects and expands the matching clones. That means we have a set of lymphocytes with us having unique receptors on that. So when the antigen is appearing, this then bind to the specific lymphocytes having that particular receptor to bind with the specific antigen. So this allows the proliferation or expansion of that particular lymphocytes, and that is known as the clonal selection process. So which upon antigen encounter, specific lymphocytes are selected, proliferated, that is clonal expansion, and then differentiated into the effector cells, that is plasma cells or cytotoxic lymphocytes, and the memory cells. So that is actually clonal selection. So our assumption should be correct. Correct answer should be the option C, that is expansion of specific lymphocytes recognizing the antigen. Okay, correct choice is option C. So let's look deep into the process of clonal selection. Here is a pictorial representation of the clonal selection process. The first one is the hematopoietic stem cells. We have the hematopoietic stem cells, which is differentiating into the various lymphocytes. The second one is the lymphocytes. The immature lymphocytes are there. Okay, we have the self antigens represented by the red colored things here. Okay, so these self antigens, those lymphocytes, immature lymphocytes, which are actually binding with the self antigens, will get eliminated. Okay, that is known as actually clonal deletion process. And after this clonal deletion, only those potent lymphocytes are selected, and we have the set of mature inactive lymphocytes here. Okay, here mature inactive lymphocytes, and this can now interact with the foreign antigens, and this foreign antigen actually select the type of lymphocyte to get expanded or proliferated. So we have a set of lymphocytes or a clone of activated lymphocytes that is actually represented by the clonal selection process. Is that clear now? Clonal selection? Okay, let's move to the next question now. Again, on clonal selection. Clonal selection explains all except option A, immunologic specificity, option B, self-tolerance, option C, antibody diversity, option D is phagocytosis. Now your thorough with what is clonal selection. So you must be able to answer this, right? First one, immunological specificity. Yes, it is offering immunological specificity. And self-tolerance. What is actually self-tolerance? Self-tolerance is the ability of the immune system to recognize self-produced antigens as non-threatening while appropriately mounting a response to the foreign substance. That is known as the self-tolerance. We have discussed about the clonal selection in the previous clonal selection as well as clonal deletion in the previous slide. So self-tolerance is all related to clonal deletion. So all the lymphocytes which bind with the self antigens get eliminated, and only those lymphocytes which actually recognize our self antigens as non-threatening will be moved to the next stage. So that is represented by the term self-tolerance. So again, clonal selection is actually representing this self-tolerance. Then antibody diversity. Yes, it is having the antibody diversity. And phagocytosis. Phagocytosis. Does clonal selection represent phagocytosis? No. So the correct choice is option D, phagocytosis. So out of the 4 choices, all the choices except phagocytosis is explained by the clonal selection. Okay, correct. So let's move to the next question. The fourth one. The process by which lymphocytes that recognize self are eliminated during maturation is. You must be able to answer this question. Clonal selection, clonal deletion, somatic recombination, energy. It is obviously clonal selection. Clonal deletion. It is deleting those lymphocytes which recognize the self antigens. One uncommon term, term is that is energy. Let's look what is energy. Energy is a state of inertness or unresponsiveness of the immune cells, particularly T cells and B cells, when they encounter an antigen. That is represented by the term energy, a kind of inertness of the lymphocytes to bind with the antigen and eliminate that thing. So our answer is clonal deletion, the way by which the self-recognizing lymphocytes are getting eliminated. Clear? Now let's move to the next question. The fifth one. A T helper cell is activated when? So options are when it binds free antigen directly via its TCR. TCR is T cell receptor. Then whether it recognizes antigen MHC2 complex on an APC with co-stimulation. APC is antigen presenting cell with co-stimulation. Option C is it binds antigen MHC1 complex on an infected cell. Option D is it secretes cytokines before antigen contact. So what would be the answer? Let's move into the detail. We have 2 types of T cells available. T helper cell, that is TH cell, which is having the CD4 receptor on that. So they are generally represented as CD4 plus T cells. They recognize the antigen which is presented on the MSC2 type, that is major histocompatibility complex type 2, which is only expressed by the antigen presenting cell. So I'm repeating the scenario. We have the T helper cell that can get activated only upon binding to an antigen that is presented by the MSC2 molecule on an antigen presenting cell. And upon this binding, it secretes the cytokines which can regulate B cells, TC cells, that is T cytotoxic cells, and macrophages. So that is how T helper cells are getting activated with the help of MHC2 molecule. And we have the second category, that is the T cytotoxic cells, that is TC cells, which is having the receptor CD8. So we represent these TC cells usually as the CD8 plus T cells, and this recognize the antigen which is presented on MHC1 molecule, MHC1 type 1 presented on the all nucleated cells. And this then differentiate into the cytotoxic T lymphocytes, that is CTLs, which then monitor the body and eliminate the altered cells through cell killing activity, that is cytotoxic mechanisms. What are these altered cells? These are virus infected or infected cells or tumor cells or the graft cells, which are all altered cells. So this will be recognized as the foreign cells and get eliminated by the TC cells. So TH cells require MHC2 representation, MHC2 molecule to get activated, whereas TC cells recognize the antigen which is presented on the MHC1 molecule. And MHC2 molecule is actually occurring in all the antigen presenting cells, and MHC1 molecule could be seen in all the nucleated cells. Okay, so now the background is clear. Now let's look again into the question. The question is, a T helper cell is activated when? So the answers are, it binds free antigen directly via its TCR. So now we know that T helper cell cannot bind directly to an antigen through its TCR. It needs some presentation on the MSC molecule, either MSC as it is T helper cell, it needs the help of MSC2 molecule for its activation. So it is not the answer. Option B, it recognizes antigen MSC2 complex on an APC with co-stimulation. Yes, it recognizes the antigen which is presented on the MSC2 complex on the antigen presenting cell. So that is the answer, option B. So now it's clear. So this is a pictorial representation of how T helper cell and T cytotoxic cells are getting activated. We have the T helper cell here on the left side panel, which is having the CD4 receptor. So with the TCR, it is binding to the antigen that is presented on the MSC2 molecule of an antigen presenting cell. So upon activation, upon this binding, it gets activated and releases various cytokines, and these cytokines can then regulate the development of, proliferation of B cells as well as macrophages. So this is how the T helper cell is getting activated. And here on the right side panel, we have the T cytotoxic cell activation. We have the immature TC cells here with the TCR on the surface. It binds to the antigen that is presented on the MHC1 molecule on all the nucleated cells and get activated. Okay, we have the mature cytotoxic TC cells here, which then eliminate the non-self cells, virus infected cells or tumor cells like that. Okay, is that clear now? How the T helper cell or T cytotoxic cells are getting activated? Okay, let's move to the next question. So the sixth question is, antigen presenting cells are uniquely required for the activation of T helper cells. So we know that thing. We just now learned about how the APCs are taking part in the activation of T helper cells. Okay. And in addition to displaying antigen complexed with class 2 MHC, what other essential component must an APC provide to fully activate a T helper cell? Good question, right? So it is asking whether, what additional requirement should be there for a T helper cell activation in addition to the presentation by class 2 MHC molecule. Is that clear now, question? Okay, the choices are a membrane-bound B cell receptor, option B, an antibody complement complex, option C, a co-stimulatory signal, option D, the CD8 co-receptor. Actually, T cell activation is a two-signal process. We can classify that T cell activation as two-signal process. Signal 1 we have discussed in the previous question, that is activation by binding to the antigen that is presented by the MSC molecule. So in addition to that signal, we have one more signal needed for the complete activation of the T cell, that is signal 2. It is non-antigen specific actually, co-stimulatory signal. We call it as the co-stimulatory signal, which actually involve the involvement of some receptor on the antigen presenting cell as well as some receptors on the T cells, which then bind in addition to the antigen presentation. So example is interaction between B7 receptor on the antigen presenting cell and CD28 receptor on the T cell, and this is required to prevent inappropriate activation or the autoimmune processes. So here let's look back to the choices available. So the additional requirement for the T cell activation in addition to the class 2 MHC antigen complex is that, which one is the answer? It is the co-stimulatory signal. We need, in addition to the MSC representation, MSC presentation, we need a co-stimulatory signal to fully activate the T helper cell. Okay, so the correct answer is option C, a co-stimulatory signal. So this is the pictorial representation. We have the macrophage here, that is the antigen presenting cell, and here you can see the class 2 MSC molecule which is bound with the antigen, and this antigen is then bound with the T cell receptor of the T helper cell. So that is the first signal happening, and together with that the B7 receptor of the antigen presenting cell is interacting with the CD28 receptor of the T helper cell. So this acts as the co-stimulatory signal, and this is essential for the T helper cell activation. And one more example has been given, B cell. We have the B cell here. B cell have the antibody on the surface on that, which is actually encountering with a foreign pathogen, foreign antigen. Together with that, there are reports that B cell can also act as antigen presenting cell to the T helper cell activation. So it is having the class 2 MHC molecule, which is having the bound antigen, and this antigen is antigen is presented to the T helper cell, okay, to the TCR receptor, T cell receptor here. So this act as the primary signal, the first signal. Together with that, the CD40 receptor of the B cell can also interact with the CD40L receptor of the T helper cell, and this act as the co-stimulatory signal for the B cell involved activation of T helper cell. Here B cell act as the antigen presenting cell. Is that clear now? Okay, let's move to the next question. Seventh question is, the ability of an organism to resist anthrax due to high body temperature represents what? First one, acquired immunity. Second choice is passive immunity. And third choice is physiologic innate barrier. And the fourth choice is adaptive immune tolerance. So, which is the correct answer? So, before going into that, let's look into the types of innate barrier. You know that what is acquired immunity? It is acquired throughout the life journey, right? Passive immunity, that is we will receive the antibodies passively, okay? We will get the antibody directly as a short-term relief. So that is represented by the passive immunity. So we will look into the types of innate barriers here. We have 4 types of innate barrier, that is inborn system to defend against the foreign pathogens. We have the first innate barrier, anatomic barrier, and within that skin and mucous membranes are the skin. We know that it is the keratinized outer layer, acidic in nature, and it acts as the mechanical barrier. And second one is mucous membranes, which trap microbes, secrete antimicrobial peptides, and act as the host commensal flora. So this is the anatomic barrier, and we have the physiologic barrier also, which is represented by the temperature, pH, lysozyme, interferons, complement, and the collectins. So that is represented by the physiologic barrier. And we have the phagocytic barrier, that is we have certain phagocytic cells within us, within our body, that is actually encountering with the foreign antigens, foreign cells. So these phagocytic barrier are specialized cells like the monocytes, neutrophils and tissue macrophages, which engulf or phagocytose the microbes into phagosomes, which then fuse with a lysosome for enzymatic digestion. So this process we have already discussed. And we have the inflammatory barrier. So that is actually involved in the inflammatory response. So this is mediated by mainly histamines, kinins, complement and cytokines, and this leads to vasodilation, increased permeability, leukocyte recruitment and fibrin deposition. So this you have observed during wound formation. When you have a cut, this inflammatory response would have been occurring immediately. So these are the innate barriers, that is inborn barriers of the defense system of our body. So let's look back to the question. The ability of an organism to resist anthrax, a foreign organism, due to high body temperature. High body temperature we have seen here in physiological barrier. So what will be the correct choice? We have acquired immunity, passive immunity, physiological innate barrier and adaptive immune tolerance. So the correct answer would be option C, physiologic innate barrier. Okay, let's move to the next question. The eighth question. Tissue damage triggers an inflammatory response. We know that whenever a tissue damage is happening, it triggers an inflammatory response. Of the Roman physician Celsus' 4 cardinal signs, the sign of dolor, that is pain, is primarily and directly caused by the activity of which chemical mediator? Okay, so out of these chemical mediators, which is actually responsible for having pain sensation during inflammatory response? So that is the simplest way, simplest representation of this question. So options are histamine, bradykinin, C-reactive protein and lysozyme. So the correct answer is actually bradykinin. Option B, bradykinin. It is a kinin peptide that is generated during the inflammatory process, and it is the major mediator responsible for stimulating local nerve endings, that is the pain receptors, and it leads to the sensation of dolor, that is pain. So bradykinin is the correct answer to this. For the pain sensation, the chemical mediator involved is bradykinin. So let's look deep into the 4 cardinal signs of inflammation that is represented by Celsus. We have the 4 cardinal signs, that is rubor. Rubor means redness of the inflammatory site. Then tumor. Tumor represents the swelling or edema of the inflammatory sites. Then calor. Calor represents heat generation. You must have observed some kind of heat generation in the affected or the inflammatory regions. You must have sensed that thing. So that is the calor. And the last sign is dolor, that is pain sensation. So the major events that is associated with the inflammatory response are vasodilation, increased capillary permeability and the phagocyte influx. So what is vasodilation? It is the dilation of the blood vessels here. So vasoactive factors like histamines, it increase the blood flow causing redness, that is rubor, and heat, that is calor. So that is the step vasodilation. And the next step is increased capillary permeability. So what is actually happening within that? It facilitates influx of fluid, that is exudate, leading to tissue swelling or edema, that is represented by the sign tumor, and serum proteins like complement and C-reactive protein. So that is the increased capillary permeability step. And last step is phagocyte influx. What is that? Influx of phagocytes to the affected area. So we have certain chemotactic factors which guide the phagocytes through the sequential steps. The first step is margination. Margination means adherence of these phagocytes to the endothelial surface. Okay. Next is extravasation of these phagocytes to the affected tissue region. So extravasation is also known as diapedesis to the tissue, emigration into the tissue, and from that tissue it has to migrate to the site of infection, and that is known as chemotaxis, migration to the site of infection. And bradykinin, a kinin peptide, stimulates the pain receptor, giving the pain sensation, and finally fibrin deposition by the clotting system. It walls off the injured area. So what is actually happening in phagocyte influx? It is the influx of phagocytes. First it adheres to the endothelial surface, extravasate to the tissue, and from the tissue site it then moves to the affected area, particularly to the site of infection. And then bradykinin, it induces pain receptors to give the pain sensation, and finally fibrin deposition occur to mask the injured area. Okay, and the final outcome is rapid containment of infection and activation of the adaptive mechanisms, and in this way we will get cure for the infected site. So this is the major steps that is associated with the inflammatory response, and we have the 4 cardinal signs also: rubor, tumor, calla, and dolor. So this is again a pictorial representation. We have an injury happening here, histamine or kinin release, which help to recruit the white blood cells, debris, and phagocytes deposition leads to healing. Then capillary leak is the edema, the 4 cardinal signs: redness, swelling, heat, and pain. Blood supply is increased, that also leads to the healing process. So just all those things we discussed in the previous thing, but just a pictorial representation for your easy understanding. Okay, so let's move to the next question. The 9th question is again related to the cardinal signs of inflammation. This you must be able to answer a very simple question. The cardinal signs of inflammation were first described by Galen, Pasteur, Celsus, Ehrlich. Who's that? Celsus, obvious, right? Previously we discussed, we said it as the Celsus cardinal signs because it is discovered by Celsus. So the answer is Celsus. We have the 10th question, last question. Which immune component directly mediates humoral immunity? We have the options: CD8 plus T cells, that is TC cells, plasma cells, macrophages, and NK cells. Which are they? Which directly mediates the humoral immunity? Obviously the plasma cells. Okay, we have the B cells, which is actually giving the humoral immunity through the antibody production, and T cells is actually giving the cell mediated immunity, that is cytotoxic and regulatory functions. So the question is which immune component directly mediates humoral immunity? So now we know that it is the plasma cells. We know that B cells upon activation will be differentiating into plasma cells and effector cells. So obviously the answer is plasma cells, which is mediating the humoral immunity. So the correct answer is option B, plasma cells. So let's look into how the B cell is getting activated to form the plasma cell. So here we have the B cell here with the B cell receptor. Upon antigen encounter, upon antigen binding, it will get activated, and in addition to that it will receive some chemical signal from the activated T helper cell that we discussed in the previous questions. Upon antigen encounter, this T helper cell will get activated to release some cytokines which can activate the B cells and macrophages. Okay, so this stimulus also increase the B cell activation. So upon B cell activation, this will form the plasma cells as well as the effector cells. Plasma cells are those which releases the antibody. Okay, so this is how the plasma cell development is happening. Okay, is that clear now? Okay, fine. So we have the reference with us. So this is all about today. We have discussed 10 important questions that covers almost all the basic areas of immunology. So I think it would be really beneficial for your future competitive exam preparations. So let's wind up for today. And yeah, see you again. Thank you.